Okay, are we ready?
Yes.
Yeah, tuko sawa.
Yes.
So we'll just go through because our lesson is supposed to start at 9, and of course, from the look of things, we still have some people who not joined the meeting. So meanwhile, I think what we'll do, we just review what we've already covered so far. So I had started talking about what we had started covering in our unit in Introduction to Microbiology. That was Unit 1. So I'll just go briefly through it, trying to tell you what is expected out of you. And of course, I presume that at least we are doing our own personal study, just to ensure that our journey is comfortable. We're not facing a lot of challenges, okay? So remember, we started our unit of Introduction to Microbiology by trying to define what is microbiology. So remember from what we had said the other time that microbiology is divided into various branches, but our major focus is majorly on medical microbiology. And we define medical microbiology as a branch of microbiology that deals with microorganisms, okay? So microorganisms, these are the invisible creatures that you can't see with our naked eye, of course, which included bacteria, virus, fungi, and parasites of medical importance. So we are saying medical importance because some of these microbes, that is either bacteria or virus, have been associated with human disease, and that's why we are terming them to be of medical importance. So when you're looking at these microbes, our major focus, of course, is trying to understand how do they cause disease. So when you're looking at how these microbes are causing disease, of course, our major focus is looking at microbial pathogenicity, okay? How microbes cause disease. Then, of course, apart from looking and trying to understand how they cause disease, we are also interested in understanding what do we see in people who have these diseases. So this is the clinical picture of a person who is either having a bacterial infection, a viral infection, or sometimes even a fungal infection. We look at what will be the manifestation. So what do we see when people have come into contact with this disease? Okay, and of course that forms the basis of our journey in medical microbiology. Then of course we also need to understand on how our body behaves when it comes into contact with these microbes. So, and the only system in our body that is associated with an interaction with this microorganism is our immune system. So our immune system is the system that will have an interaction with either bacteria or a virus, okay? So just to define microbiology in a simple terms, we just say microbiology is the study of microbes. And in this context, microbes are the small microorganisms that we are talking about. And remember, the microorganisms are the 4 groups of microbes, that is either bacteria, a virus, a fungi, or sometimes a parasite, okay? Then, of course, we also need to understand what happens. So you come into contact with the bacteria, you come into contact with the virus, you come into contact with the parasite. What does the parasite do? What does the bacteria do? So, of course, there's a process, there's something that needs to take place. So the same thing people are talking about coronavirus, and I'm very sure everybody is curious to understand what happens if you come into contact with coronavirus. So there are so many things that might happen. So one of it is that when somebody comes into contact with a microorganism, there's a possibility that the microorganism will not be able to establish itself within their body. So when you're talking of it not being able to establish within the body, then we are meaning that our immune system is able to eliminate it before it causes any damage. So remember, we've just talked about an interaction, and what our body uses to interact with this microbe is majorly the immune system. So sometimes we have infectious conditions that go unnoticed just because our immune system was able to clear the microbe before we even noticed that we had come into contact with something that was dangerous. Okay. So sometimes this mechanism, our immune system can be unable to eliminate, or it can be overwhelmed by the microbes. Then what happens? The microbes then will start multiplying, and of course, as it multiplies, it colonizes our body surfaces. So if it's a microbe on the skin, it will multiply and ensure that it's covering the entire surface of our skin. If it's in our mucosa, the same things applies. And this happens especially when our immune system is unable to clear the infection completely. So here they will colonize most of the body surfaces. Then of course, in itself, that in itself does not mean that our immune system just relents and stops functioning. It will still continue trying to see whether it can eliminate the infection. So however much the microorganism might continue to colonize our body surfaces, remember the immune system is also trying to ensure that it's trying to eliminate them. So there are those microbes that will eventually form a permanent relationship with the host, where they have a mutual relationship. They form what we call a transient state of colonization. Okay. Sometimes those individuals where there's continuous colonization, the individual might eventually become what we call prolonged carriers. And I'm very sure most of us are familiar with the conversation we are having with COVID-19. So there are some studies that are coming out of China where they're saying that those people who had healed from COVID-19, they eventually end up becoming reinfected with the virus again. So that one tells you that there's a possibility that the virus are not being cleared completely out of the system of the patient. So what happens? These people, however much they've gotten healed, they still become potential. They become potential carriers who can transmit the infection to other individuals. Okay. Then, of course, we also looked at how do microorganisms cause disease. Then, of course, under this part we talked about the two theories. So we had the theories of spontaneous generation that was eventually dismantled, and then, of course, we have the germ theory. And of course, the germ theory is a theory that was taught by where he tried to explain how microorganisms have been associated with infection. Then, of course, to demonstrate that, he had to do an experiment, and an experiment he tried to use food spoilage as an example of trying to say that microorganisms could be some of the things that are associated with human disease. And of course, here you see he had to make some broth, of course, this some form of food. Then when he covered the food, he realized after keeping the food for some time, the food remained fresh, irrespective of staying for some time. But when he re-exposed the same brothel or the same broths to the environment, he realized that the food eventually became spoiled. And of course, that explained that we had some microorganisms from the environment that got into the broth and eventually fermented the broth. And of course, in itself, that caused the spoiling of the broth. Okay, so it's the same thing we are talking about. People coming into contact with somebody who's having COVID-19. So as you come into contact with somebody who's having COVID-19, you eventually inhale the droplets, and when the droplets gets into your system, what is manifested is the condition that we see with those people who've been known to be having COVID-19, where there's in elevated temperatures, there's dry cough, there's diarrhea, and with the eventual development of pneumonia. Then, of course, we looked briefly at some of the contribution of some of the scientists who are involved in this field of microbiology. So we looked at a contribution made by Anton Lewandowski, and of course we said that he's the person who was the first person to discover or to invent a microscope. And of course, a microscope is an important equipment in microbiology that we use to visualize some of these microbes. Then we also have some contribution from Louis Pasteur. Of course, we just talked about him coming up with a germ theory that tried to explain how microorganisms have been associated with disease. Of course, he also had made some other contribution, something that I think you also have time to ensure that you are reviewing to understand what were some other contributions that he made, especially in the field of medicine. Then we have the Robert Koch. So Robert Koch is also a scientist who also had an immense contribution in microbiology. And some of his contribution, we are saying that he's the person who discovered the bacteria called Bacillus anthrax, the one that was presented by the group of Kim, okay? So that bacteria was discovered by Robert Koch when he was trying to advance the knowledge on understanding how microorganisms have been associated with infection. So he also discovered the MTB, the Mycobacterium tuberculosis, the one that is associated with TB. Then he's also the one who also discovered the bacteria Vibrio cholera, the one that is associated with cholera. So apart from discovering some of the bacteria we know of today, he also came up with what we call Koch's postulate. And within Koch's postulate, he wanted to explain how do you link a microorganism to an infection. So he came up with the 4 postulates. So where he said that the first postulate, he said that for you to say that a given microbe is associated with a given disease, then we need to be able to isolate that given microbe from the patient. Okay, so then of course, after isolating it, then if you inoculate the microbe into another animal, the microorganism is supposed to cause the same disease. So which means if the microbe is associated with either cough or diarrhea, then we need to see the new animal or the new host exhibiting the same characteristics, okay? Then of course, we need to re-isolate the same microbe from an animal again. Then of course, if we are able to do that, then that we explain and it helps us to say that a given condition is associated with a particular organism. So if I was to talk of the COVID-19, of course, that's what is happening currently in the world. So for us to say that you have COVID-19, then we need to isolate the virus from your system. So unless we do that, then we cannot say that you're having that particular microbe. Then we also looked at the contribution of Joseph Lister. So we say that he's the father of the current antiseptic surgery. So he's the one who brought about the aseptic procedures that you are trying to practice within your clinical practice, especially in theater. So, and of course, it's the same concept we are trying to embrace now. So when you're telling people wash your hands, cover your mouth with a surgical mask. So remember, this is some of the concepts that were initiated by Joseph Lister. So however, some people don't embrace them, but for him, he realized that they had an immense contribution, especially in regarding reduction in nosocomial infection. Because by that time, people were not taking the necessary precaution, and they realized that whenever a patient went to theater, they came out with an infection. So, and of course, to improve that, is the one who initiated the current aseptic procedures that we undertake when we are carrying out our procedures in theater. okay? Then we looked at now the specific microbes. So we say that our microbes can be divided into two broad categories, looking at their cells. So we say that most of the pathogenic, and of course these are the bacteria, they are called prokaryotic pathogens because they are, if you look at the structure of their cell, lacks a nucleus. So that's something that you need to comprehend and understand. So remember, all bacteria are prokaryotic cell, and whatever makes them become prokaryotic cell is just because their cells are made up, or the genetic material is not enclosed in a nuclear membrane. So in simple terms, we think that they are non-nucleated, so they don't have a nucleus. Then of course we have the other group of microbes. Those are like, for example, the fungi, the protozoa, the helminths. Those are called eukaryotics. So they're eukaryotic because they are the cell structure, they have a nucleus with them. And of course, if you are to compare them with our cell, then we say that they have a closer resemblance to a human cell. Because remember, a human cell also, they also have a nucleus plus other cellular organelles. Okay. Then we looked at briefly at some of the characteristics of a bacteria. So and some of the things we looked at is the shape of bacteria, where we said it can either be a rod, spherical, or screw-shaped. Okay. Then we also say that bacteria also has a cell wall, and of course that is a distinguishing feature if you compare between a human cell and a bacteria cell. So a bacteria cell has a cell wall. Our human cells do not have a cell wall, and of course that is the same advantage we've actually capitalized, especially when we are using the antibiotics that actually target structure. And of course, because we don't have a cell wall, then our antibiotic is able to deal with the bacteria without bringing a lot of damage to our host tissue. Then we also say that they also divide by binary fission. All bacteria, they divide through the process of binary fission. And remember, binary fission, we say that the cell divide, a mother cell divide to give us two daughter cells. Okay. Then, of course, we also looked at fungi. So remember, this was just a general characteristics of microbes. So we also looked at some of the characteristics of fungi. So where we say that they are non-photosynthetic. We also say that they are saprophytic, which means they live on dead matter, and at the same time they're eukaryotic. So which means that within their cells they have a nucleus. So looking at their morphology, we say that they exist into two forms. So we have those ones that are either unicellular, or sometimes they form what you call filaments. Okay. So unicellular, which means they are made up of a single cell, and when they become filamentous, which means their structure has got more structures attaching to each other. Then looking at their reproduction, given that they're living things, so we say that fungi also reproduce either asexually, sexually, or both. So when you're talking asexually, it's where there's production of young ones without involvement of gametes. But if gametes are involved, then we say that is an example of a sexual reproduction. Then, of course, because they are fungi, we have some fungi that have been associated with the human disease. Then we are also saying that within that group, we have fungi that are pathogenic to human beings. And of course, that is covered, especially when we'll be looking at our unit in mycology. So, of course, where we'll be looking how do fungi cause disease. Then we have the protozoa. So protozoa, these are also still eukaryotic microorganisms. So, and of course, looking at their characteristics, we are also saying that the protozoa are single cell. So which means their body is made up of a single cell. They're not multicellular. So they're also non-photosynthetic. So which means they don't undergo photosynthesis because, of course, they lack the important element, the one we call the chlorophyll. Okay. So then, of course, when you look at their living style, we also say that they are free living. So we have those ones that have free living lifestyle. Then we have those ones that have what we call a parasitic lifestyle. So a parasitic lifestyle in the sense that they depend on other creatures for nourishment. But not all parasites or not all protozoa are parasitic. We have those ones that can live freely in nature. Okay. Then we also say that most of the protozoa infect various tissues in our body. So there are those protozoa that you'll find within the liver, there are those that you'll find within the blood circulation and so forth. Okay. Okay. Then of course we also looked at the general features of helminth. So helminth is also an example of a parasite. So we say that helminth are the worms. And one distinction feature of a helminth, if you are to compare it with a protozoa, is that most helminth are multicellular. So which means their body structure is made up of more than one cell. So they are also still eukaryotic cell, so which means their cell is made up of a nucleus. Then we are also saying that they also have a complex body organization. So complex in the sense that they have respiratory system, they have digestive system, and some also have a reproductive system that is separate. And of course, we'll understand more about them when we'll be looking at the unit on medical parasitology. So under the helminth, of course, we have the groups, we have the tapeworms. So scientifically, we call them the cestodes. We have the flukes, that is the trematode. Then we have the round one, and of course, these are the nematodes. So they are called parasites just because they rely on their host for nourishment. So like, for example, if you're having roundworms within your tummy, what the roundworm does, it gets all its nourishment from you. So as you take your food, they also take advantage to have the same food with you. And of course, that's why most helminth have been associated with malnutrition, depending with the type of the helminth. Then finally, we looked at the viruses, and of course, I'm very sure we need to put a lot of interest in this because we are online just because of our virus. Okay. So some of the characteristics we see in viruses is that most viruses, or absolutely all viruses, are obligate intracellular. So obligate intracellular in the sense that they live inside a cell. So which means they cannot survive outside a cell. Okay. So they are called obligate intracellular because they have absolutely dependent on their host cell for survival. So looking at the genetic material, we say that a virus can have either a DNA or an RNA, and they cannot have both. So, and it's the same molecule, that is the genetic makeup we sometimes also use to classify them. So we can either say this is a DNA virus or this is an RNA virus. So the same way we are talking about COVID-19 being an RNA virus because its genetic material is made up of an RNA. So then, apart from its genetic material, there's also another structure that is found on a virus. And of course, this is what you call the capsid. So the capsid is the protein code that encapsulates the genetic molecule of a virus. So if a virus is having a DNA strand within itself, then it will eventually have a protein code around it to protect its genetic information. So apart from that, we also see that we have some viruses that have what you call an envelope. And I'm very sure all of you are aware when you're talking about the virus that is currently bringing trouble, we are saying that it has an envelope, and that's why we are encouraging people to hand wash. And the concept is that when you do hand washing using a soap, the soap will eventually destroy the envelope. And when the envelope is destroyed, then it renders the virus to become become useless. And of course, that's why we are emphasizing that people need to hand wash. And just because this virus has got an envelope on its surface. And of course, when you'll be looking at the unit of virology, will understand what is the role of this envelope, or what makes these viruses become more liable when they're exposed to heat as compared to the naked virus. Okay. Hello? Then we also say when you're looking at a virus structure, we are saying that a virus will only replicate within its host cell. And the reason why a virus will replicate only within its host cell and taking this obligatory intracellular lifestyle is just because if you are to look at a virus structure, we say that a virus structure is only made up of at least two structures. So we have the protein coat, the protein coat. This is the protein coat. Then within the protein coat, we'll be having its DNA. I wish I was good at drawing. Okay, so within it we have the DNA and the protein coat. So for a normal cell to function, it's supposed to have other structures for it to function. For example, it's supposed to be having a ribosome. And of course, we understand ribosomes are meant or are there to help the cell manufacture protein. So because a virus does not have these structures and it requires to manufacture its protein, then you find that it will need to rely on its host cell to manufacture its protein. And that's what makes all viruses to have an intracellular lifestyle. Okay. So and then of course, because these viruses have got an intracellular lifestyle, and then of course, the fate of the host cell is under the mercy of the infecting virus. So once the virus enters the cell, then of course it takes over all the cellular processes within the cell. And this is what makes the cell either die by the process of cytolysis, or it might die because it's unable to function normally. And of course, it's eliminated by the immune system. Okay. So then, of course, that was a summary on how to differentiate between a prokaryotic cell and a eukaryotic cell. So what I want you to remember here is that the absolute distinguishing factor is the presence or absence of a nucleus. So a cell is prokaryotic because it lacks a nucleus, and a cell is eukaryotic because it has a nucleus within itself. Okay. So do you have any queries? Because I think we need to stop here and continue with our normal lesson. Because I see we are almost around 22. Any question, Nancy? Nancy?
Yeah, no, thank you.
You have any question? Tabby? Tabby? Tabby?
No question.
No question. Okay. So I think what we'll do next week, we'll still continue with our remedial revision. So we'll start from here, but remember we also need to continue with our normal lesson. So remember, first thing in the morning, I'll give you an opportunity. We go through what we've learned. If there's any problem that need to get some clarification, we'll do that during that process. So remember, we'll say that we'll be having our remedial from 7, maybe all the way to 8 as we wait for the rest of the team to come on board. You think this will benefit you? you are free to join the conversation. So as you go through your revision, if there are some things you're finding some challenge with, just note them down. Then maybe when we have the remedial session, we can have the discussions together. Is that right?
Yes.
Okay.
Okay. Okay, somebody to remind us what we covered last week, because I'm told there's a person who didn't manage to hear what we discussed last week. Gladys? Gladys? You unmute yourself. Nancy?
This area, but
Can you share with us what we covered last week? Nancy?
Part of what we shared include the gonococci
Yes.
and listeria. So let me start with the listeria.
Yes.
You talked about it's a slender, short, gram-positive rod bacteria and do not form spores.
So listeria was associated with what condition?
Listeriosis.
So listeriosis. So
yeah, which affects usually mainly pregnant women and also food, if I'm not wrong.
Yeah, so that is the mode of transmission. So you get the infection when you eat contaminated food, okay?
Yes.
Yes. And then Tabby. Tabby? Tabby. Caroline. Caroline, are you there?
yes.
Okay, so from the way Gladys has told us last week, we looked at Listeria monocytogenes. Then we also looked at Neisseria, the species, and of course, we looked at Neisseria gonorrhea. So what did we say about Neisseria?
About Neisseria, we said
yes,
which is common for causing meningitis and gonorrhea. Type of disease, pyogenic typhoid produces pus. Neonatal newborns, prosec, and Neisseria sorry, bacteremia, and then also an anal from a result of having anal sex.
So we had covered on Neisseria gonorrhea. So we said under the Neisseria species, we have the two species, that is Neisseria gonorrhea, which is a sexually transmitted infection. Although sometimes it also causes infection, especially in the neonates, especially if the mother was having Neisseria gonorrhea. And of course, that informs the reason why we always do urinalysis, especially to pregnant mother, to see whether they have any sexually transmitted infection. Then, of course, we also looked at some of the drugs we use to manage Neisseria gonorrhea. Eliud.
Yes.
An example of an antibiotic we said we can use to manage gonorrhea?
We mentioned about penicillin.
So we talked about penicillin. Hello? Yes, yes.
Ceftriaxone.
Yeah.
And doxycycline, which we said it is effective against chlamydia.
Or chlamydia. So remember, Neisseria gonorrhea causes urethritis, that is inflammation of the urethra. So apart from gonorrhea, we also have chlamydia that also causes urethritis. So sometimes it's also good to do a conservative management, especially if you don't know what is the cause of the urethritis. Okay, so we give doxycycline just to cover the patient in the event they have chlamydia infection. Okay. So for this week, we'll continue. So, but I presume that everybody is having an opportunity to go through the slide because we need to move very fast so that we ensure that we are covering whatever we need to cover during this session. Okay. So the other, do you have a question? Gladys? Okay, so I will request kindly, if you're not speaking, mute your mic so that it doesn't cause the background interference that we are hearing. Okay, thank you. Gladys, do that. Mute your microphone. Okay. Okay, so the way I was saying that under the Neisseria species, we have the two species, Neisseria meningitis and Neisseria gonorrhea, and we've just said that Neisseria gonorrhea is an example of a Neisseria bacteria that is associated with sexually transmitted infection. So there's this other species, the one we're calling Neisseria meningitis. So just from the name meningitis, that one tells you that these are an example of a bacteria that causes meningitis. So meningitis in itself, remember, is coming from the word meninges. Meninges are structures or is a membrane that is found or that covers our brain. So when that membrane is inflamed, either because of an infectious process, then we term that inflammatory process to be meningitis. So when you're looking at meningitis, remember this is a general term. It can either be a bacterial meningitis, viral meningitis, or sometimes even fungal meningitis. And of course, we'll understand them when we'll be looking at the other groups of microbes, especially we have those viruses that are associated with the meningitis. At the same time, we also have some fungi like the Cryptococcus neoformans that is associated also with the meningitis. So Neisseria meningitis is an example of a bacteria that causes inflammation of the meninges. Then we also saying that it's an example of a bacteria that has been associated with epidemic. So remember in your reflection, I had asked you to give a reflection on what is happening and trying to relate it with a bacteria pandemic or epidemic. So if you are to look at examples of bacteria that have been associated with epidemic, Neisseria meningitis is an example of a bacteria that causes a certain upsurge of the number of cases. So, and majorly, it's been associated with outbreak, especially in institution. And the institution could be either school, prison, or sometimes even in the military. And the reason why there's always this outbreak is just because, remember, these are examples of areas where there's a lot of congestion. And remember, when there's a lot of congestion in itself, this gives a harmful opportunity for the bacteria to spread within a given population. Gladys, kindly mute your microphone. Okay, thank you. Okay, so let's continue. So looking at their general characteristics, we are saying also Neisseria meningitis are non-motile. Gladys, mute your microphone, please. Okay, so we are saying that looking at their general characteristics, we are saying that they are non-motile. And the reason why we are saying that they are non-motile is just because they don't have a flagella as a structure or on their cell, okay? Then we are also saying that they are gram-negative diplococci. So which means they have similar shape if you are to compare them with the Neisseria gonorrhea. So they are gram-negative, which means they don't take the purple color when you stain them with gram stick. So we are also saying that when you look at their shape, they also have a bean-shaped or a kidney-shaped like structure. And of course, it's the same thing we also talked about when we are looking at the Neisseria gonorrhea. So if you are to look at the shape of Neisseria meningitis and Neisseria species, looking at their shape, you cannot be able to distinguish between the two, just because they have some form of resemblance, whereby they are either forming diplococci structures or they take a bean shape like appearance, okay? So they also have pili, a similar characteristic that we also saw when we are looking at Neisseria gonorrhea. And of course, we say that the function of the pili is to enable the bacteria attach itself on our body structure. So for Neisseria, we say that it attaches itself within the epithelial cell of the urethra. But for Neisseria gonorrhea, because it's associated with the meningitis, then it's supposed to attach itself within the epithelial cell within the nasopharyngeal mucosa. And of course, we say the reason why this bacteria is supposed to have this pili is to enable the bacteria colonize those surfaces. Because in the event they lack this pili, then they can easily be washed away by urine, if it's the case of Neisseria gonorrhea. And if it's for the case of Neisseria meningitis, remember within the mucosa, there's continuous flow of mucus. So without having a pili or a structure that will enable the bacteria attach itself, then of course, because there's continuous circulation of mucus within the mucosa, then they can easily be washed away by the mucosa, okay? So looking at its epidemiology and pathogenicity, so we say that people get infected by Neisseria meningitis when they inhale droplets from a person who is harboring the bacteria. And of course, this is just similar to the same thing we are talking about COVID 2019. Can I request those people who are having their mics on to mute them, please? just look at your mic where you see an icon of a mic and unmute it, please. Techno, mute your microphone, please. Okay, so we are saying that the mode of transmission people get in Neisseria meningitis, or sometimes get bacterial meningitis when they inhale droplet. And of course, here we expect the droplet to be having the bacteria. And then, of course, once the bacteria is inhaled within the mucosa, we say that the bacteria will attach into the nasal mucosa lining by use of pili, where they will eventually produce enzyme, then find their way into the system, then spread from the mucosa through the bloodstream, then to the meninges where they cause inflammation. Then, of course, here we say that the incubation period is between 2 to 10 days. So from the time of exposure, it will take an individual between a period of 2 to 10 days before they start exhibiting the signs of having bacterial meningitis, okay? So we are also saying that they also have some antiphagocytic property. And remember, when we are looking at our unit in immunology, or when we are looking at some of the structures we found in a bacteria, we see that we have some bacteria that are equipped with what we call a capsule. And a capsule is a virulent factor that enables a bacteria evade the process of phagocytosis. So Neisseria meningitis is an example of a bacteria that has a capsule, and this capsule is meant to protect the bacteria from being phagocytized. So remember, for it to cause infection, it's supposed to find its way through the bloodstream into the meninges, which are located in the brain. So for them to pass through the bloodstream, remember, within the bloodstream, we have what we call the white blood cell, and an example of white blood cell are the phagocytic cell. So how does it then pass through this bloodstream without being noticed or being destroyed by the immune system? So what they do, they equip themselves with a capsule that will enable them, or will aid them travel all the way from the nasal mucosa up to the meninges, where they cause an infection. So we also say that they also produce another substance, the one we are calling IgA protease. So protease is an enzyme that degrades protein. So remember, within our mucosa, our immune system protects us by producing immunoglobulin A. So because we have immunoglobulin A all over our body surfaces, and for that matter, within the mucosa, we are saying that Neisseria meningitis carries an enzyme that destroys our antibodies. So for it to colonize the surface, it's supposed to get rid of this immune mediator so that it can be able to cause an infection. So it having a protease, which is an enzyme, this enzyme will eventually destroy this molecule, that is the immunoglobulin that are found within the mucosa. Then, of course, once the immunoglobulin A is destroyed, then our mucosa becomes vulnerable and the bacteria is able to colonize, okay? So the clinical significance or the clinical picture we see with somebody is having Neisseria meningitis. We are saying that Neisseria meningitis causes meningitis, and of course this is something I've already explained, where once they penetrate the mucosa barrier, they enter into the bloodstream, and of course multiplying within the blood circulation, then they find their way into the brain, where they cause this condition we are calling meningitis. So some of the clinical features you see with somebody is having Neisseria meningitis. An example will be this patient will complain of having severe headache. They will have a rigid neck. Some will vomit, and sometimes some will have what we call, they will have sensitivity to light. So which means their eye will not be able to tolerate any form of light. So, and I'm very sure these are some of the conditions we've come across during our clinical practice. So where a patient comes into the clinic is having complained of having severe headache, and then of course their neck is rigid, which means they cannot bend their neck. It's just because of the inflammation that is taking place within the meninges. Then of course they become photophobic, so which means they will not have light. So if a patient comes with such a characteristic, then there's high likelihood that this patient could be having or could be suffering from Neisseria meningitis. So if this condition is not managed on time, this might lead into coma. And of course, once a patient is having meningitis leads into coma, and then it becomes very difficult for that patient to recover. So it's always good, especially for you nurses, especially when you're taking your clinical history, you need to put a lot of consideration to understand what could be the patient complaint of having a severe headache be linked to. So there's most likely that the patient could be having a bacterial meningitis. So, and of course, you need to take the necessary steps to ensure that the patient does not slip into coma, because if you let the patient slip into coma, then there's a high chance that we might eventually lose the patient. Then, of course, other conditions associated with the Neisseria meningitis are septicemia. And of course, septicemia is where we're having the Neisseria meningitis within the blood circulation. And of course, them being in the blood circulation, of course, this initiate an inflammatory process within the blood circulation where the immune system try to react to the bacteria, and of course, by producing cytokines. So, and of course, you see patients who are having who are having septicemia, they will eventually always complain of having fever and so forth. Okay. So, and then how do we diagnose them? So, laboratory diagnosis is of course by demonstrating the bacteria within the CSF. So of course we get CSF from a lumbar puncture, then we take it for microbial analysis. So what they see in the laboratory is, of course, after doing a gram stain, they will see the gram-negative diplococci we just talked about. Then of course that in itself will tell us that the patient could be having Neisseria meningitis. Okay. Then of course we can also be able to treat the carrier people. Those are what we call the reservoir host. So these are people harboring the bacteria, but themselves are not exhibiting any clinical symptom. So for such a people, what we can do what we call an nasopharyngeal swab. Then after we've done the nasopharyngeal swab, of course, we are able to isolate Neisseria meningitis in a chocolate culture treatment. So we are saying that for Neisseria meningitis, remember what I've just said, if we don't take the necessary steps as is required, our patient might eventually lead into coma. And once a meningitis patient leads into coma, then there's a possibility that we will not be able to save the patient. So any bacterial meningitis is a medical emergency. So this tells you that when you do your physical examination of a patient and you realize the patient is having a rigid neck, then you need to suspect the patient to be having a bacterial meningitis. Then you need to initiate a prompt management, because if you don't do that, there's high chance that you'll eventually lose the patient. Okay. Then of course, because the bacteria will find their way into blood circulation, and I've just said that it might lead to septicemia. And some of the symptoms or signs and symptoms we find with people having septicemia is that they will always complain of high fever. Okay, so if they are having high fever, then of course we need to take care of the high fever, where we are required to give antipyretic to try and bring down the fever. Then some of the antibiotics we use to treat for a person who is having Neisseria meningitis is an example cefotaxime, or ceftriaxone. So of course, you find that the management is also similar to the way we are managing Neisseria gonorrhea. Okay, and then we also have some other things we can do, especially on prevention and control. So sometimes we can give vaccine, especially to protect the vulnerable groups, especially those people who are in crowded areas, the way we talked of schools, or sometimes in prison, or even in barracks. So we can use a vaccine to protect the general population so that they don't get infected by this bacteria that causes meningitis. Then we can also give rifampicin or rifampin as a prophylactic measure. Like for example, you have a patient that has come to the hospital with bacterial meningitis. So remember, we say that people get Neisseria meningitis when they inhale droplets that are having bacteria. So there's high likelihood that the bacteria can also be spread to people who are found within the vicinity of that given patient. Then how do we protect those members that are in close proximity with our patient? So we can do what we call a prophylactic management, where we give rifampin to protect those individuals so that they don't eventually develop develop bacterial meningitis. Okay, so this is just a summary of the Neisseria disease. So we've talked about Neisseria gonorrhea being associated with the sexually transmitted infection. Then, of course, we concluded by looking at Neisseria meningitis, the one that is associated with the bacterial meningitis. Okay. Do you have a question on Neisseria meningitis? Any question? Anne?
Yes, I'm asking.
Yes.
You've talked about giving we've talked about a vaccine for 11 to 55 years.
Yes.
What about those people who are in maybe in an epidemic area and who are less than 11 or over 55 years? What do you give?
Mm-hmm. So remember, our vaccine here, we are using a vaccine to protect the vulnerable groups. And when you're looking at the vulnerable groups, of course, we say that some of the predisposing factors of vulnerable groups are people who are found in crowded places. And of course, we've given examples of children in school. And I'm very sure that should be the same thing we are doing here. So we encourage that all children who are going to school, they need to be given a gonococcal vaccine. And the reason why we are giving them is just because these are the vulnerable. And of course, you need to differentiate between the extreme ages and whatever. So our predisposing factor here is the environment. So people are found in crowded environment, we find that there's high likelihood that they might get a bacterial meningitis. Is that clear?
It's all right. But is there any effect if you give the less than 11 years?
Sorry?
Are there any negative effects if you give to the less than 11 years?
Less than 11 years?
Yes, because these kids still are also in school, they are overcrowded.
Okay, of course you need to ask yourself, why do we give vaccine?
Preventive purposes.
Yeah, so remember, we give a vaccine to prevent a disease. But remember, when you're giving vaccine, sometimes you also need to put into context the subject you're talking about. So one thing you need to ask yourself, what is the type of bacteria, what is the type of vaccine are we giving? Are we giving a live vaccine, or are we giving a killed vaccine, or are we giving subunit, which are extra? So live vaccine is where we need to handle it with a lot of caution, because especially to those people who have the extreme ages. And of course, the extreme ages, we are talking of people below 5 years and people above 65 years. So the reason is, of course, these people, the immune system is a little bit down, or they have a state where the immune system in an immunocompromised state. So if you are to give a live vaccine, then there's a possibility even a live vaccine, irrespective of it not being virulent, it can eventually still cause symptoms and cause disease in those individuals.
Okay.
Yeah, so remember when you're giving a vaccine, you need to give it in the context, looking at the vaccine. Is it a live vaccine or is it not a live vaccine? And I'm very sure that's the same thing we do when we're giving polio, at the same time when you're giving BCG. So we tell mothers they need to be watchful. If they realize anything abnormal, we tell the mother to bring back the baby to the clinic for the obvious reason, because those are live vaccine. They're live vaccine. Okay.
Okay.
And then I'll request that you be muting your mic.
Thank you.
I see your mic is on. Okay. Any other question? Okay, so if there's no another question, maybe we can go to the next group of microbes. So the next group of microbes, of course, it contains a group of microbes that we generally call them gastrointestinal gram-negative prods. So all this group of bacteria are found within the gastric mucosa or within our GIT. So some of the characteristics we find with this group of gram-negative rod, we're saying that they contain what we call a lipopolysaccharide structure within their cell wall. And of course, we'll understand them because remember we talked about bacteria producing toxin, which can either be exotoxin or endotoxin. So this group of microorganisms are example of bacteria that produces what we call an endotoxin. And the endotoxin, of course, is coming from this structure we are calling the lipopolysaccharide, which is found within the cell wall. So, and of course, this forms the basis of the virulent factor. So this toxin, once it's produced, is the one that brings about the fever or the pathology that is exhibited with people who are having this type of infection. So we also think that they are generally gram-negative rods belonging to different taxonomical groups, and of course we'll be looking at them. Then we get human beings get infected when they come into contact with fecal contaminated either food, water, or something like that. So remember here we are talking of this type of bacteria being found within the gastrointestinal mucosa of a human host. Okay, so examples of these gram-negative rod that have been associated with our GIT, we have the Escherichia coli, we have the Salmonella, we have the senior species, we have the Campylobacter, Shigella, Helicobacter, the one that causes H. pylori. We have the Vibrio, the one that causes Vibrio cholera. We have the Enterobacter, Klebsiella, the Proteus, and so on. And I'm very sure most of you will be asking themselves, why are we looking at Escherichia coli as an example of a gram-negative rod that is associated with gastrointestinal condition, irrespective of us saying that E. coli is an example of a normal flora. Of course, that's something we are about to understand, okay? So we'll start looking at E. coli. So remember E. coli, from what we had said initially, we said that E. coli is an example of a normal flora of the human colony. So a normal flora in the context that it does not cause disease, especially when it's found within the colony. But remember, we also say that a normal flora is a normal flora if it strictly resides within its designated anatomical site. If a normal flora changes anatomical site, it eventually no longer becomes a normal flora. And that's why we are looking at E. coli as a pathogen, because E. coli has been associated with some human disease. And some of the conditions associated with the human disease where it can cause what you call urinary tract infection. So if fecal matter gets their way into urethra, and the urethra is colonized by E. coli, it will no longer be a normal flora, but it will eventually become pathogenic. And E. coli is an example of a bacteria that has been associated with nosocomial infection, especially in our clinical practice, especially when you are doing catheterization. If you don't do catheterization the way you're supposed to do it correctly, by following this aseptic technique, you might eventually introduce E. coli into urethra of the patient, where it will eventually cause urinary tract infection. Okay. So E. coli, apart from causing urinary tract infection, we have some strains of E. coli that have been associated with the gastrointestinal infection, and of course, we'll try to understand them. Okay. So, and of course, each and every strain of E. coli is associated by producing a given type of virulent factor. And of course, this virulent factor is the one that makes the E. coli become more pathogenic. So we also say that E. coli, they have a fibrin, and of course, the same thing we've just talked about the Neisseria. So we say that they have pili, and the function of the pili is to enable the bacteria adhere itself within the mucosa. So remember, E. coli reside within the mucosa, and within the mucosa is where we have our digestive system. And within the digestive system, remember, there's continuous flow of food and so forth. So for bacteria to survive in such an environment, then it's supposed to have structures that will enable it attach itself on the surfaces. Okay. So we are also saying that they are motile. So motile in the sense that they have flagella on their outer surfaces. So which means if you are to put a colony of E. coli on a microscope slide, then we are able to see some form of motility, where the bacteria will be moving from one place to the other in the microscopic field. But we are also saying that not all E. coli are motile. We also have some strains that are non-motile, and of course, we'll be able to understand them. So looking at their fermentation characteristics, we're also saying that they are lactose fermenters. And of course, this is a feature that we use, especially when you're trying to isolate microbes in a stool culture. So remember, because they're normal flora, then how do we differentiate between an E. coli, which is a normal flora within the colony, and the pathogenic bacteria like the Salmonella or Shigella, which are also found to be associated with the infection within the colony. So the distinction feature that we normally use is their ability to ferment lactose. So because they're able to ferment lactose, Salmonella is unable to ferment lactose, then we can be able to differentiate between a colony of E. coli and a colony of Salmonella. Okay. So looking at their clinical significance, so we'll be looking at disease condition that are associated with the E. coli, especially in the intestine. So we have various strains of E. coli that are associated with the infection of the GIT. So, and of course, remember, you need to bear in mind that E. coli is supposed to be a normal flora. But we are saying that within the E. coli, we also have some strains that are not, they have the property of causing disease. And of course, those strains are the one we are mentioning here. So we have the first strain. So we have what we call the enterotoxigenic E. coli. And of course, it's enterotoxigenic because it produces a toxin within the lumen. And of course, once the toxin produces, then of course, that brings about the diarrhea that we see with the people who have infection because of this strain. Then we have another species, the one we are calling enteropathogenic E. coli, enterohemorrhagic E. coli, and enterohemorrhagic, and then we have the enteroinvasive E. coli. Then finally, we have the enteroaggressive E. coli. So remember, these are the strains that are that are pathogenic, especially in the human body. So if you have to use an example of enterohemorrhagic E. coli, the reason why it is pathogenic is just because it is associated with acute bloody diarrhea. So if a patient complains of having a bloody diarrhea and sometimes with abdominal tenderness associated with the fever, then there's high likelihood that they could be having an infection as a result of coming into contact with the enterohemorrhagic E. coli. So in this context, it's no longer a normal flora, but it's a strain that is pathogenic to human beings. So the same applies to enterotoxigenic E. coli. So for those people who are having this strain, of course, which is very common with people who are traveling, and I'm very sure most of us have had such experience. You travel from Nairobi going to Bungoma, then when you reach Bungoma, you start having a diarrhea. So what causes that diarrhea most of the time is it's been associated with E. coli, especially this strain that we are calling enterotoxigenic E. coli. So, and majorly its mode of transmission is either getting the infection through contaminated food or contaminated water. And that's why sometimes we encourage people when you're traveling, ensure that you don't eat on the road or you don't drink water in some kiosks on the road without knowing whether the water is clean or it's not clean. So if you don't do that, then you'll eventually develop an infection as a result of enterotoxigenic E. coli. So it's being called enterotoxigenic because it will produce toxins within the lumen of the colony, and of course, this in itself will lead to diarrhea. Then we have the enteropathogenic E. coli, which is a common E. coli that is especially found in young children, especially where mothers are not putting a lot of attention into their personal hygiene, or they're not practicing good sanitation practice. So, and of course, I'm very sure most of us are familiar. For those of you who've had an opportunity to have a baby during the first or during the infancy stage, young children experience a lot of diarrhea. And one of the causes of those diarrhea is just because of this bacteria or this strain of E. coli we are calling enteropathogenic E. coli. Then, of course, you have the enterohemorrhagic E. coli, and of course, this has been associated with production of lesion within the colony. So they produce an enzyme that digests the mucosa within the colony with an eventuality where there is bleeding within the colony. So they have some resemblance to Shigella, and of course, you'll understand what are some of the characteristics of a person who is having Shigella. So they produce Shigella-like toxin within the lumen, and it's the Shigella-like toxin that will bring about changing in the dynamics within the lumen of the colony that will eventually lead into a bloody diarrhea. So then you have the enteroinvasive E. coli. So this also gives a dysentery-like symptom where the patient will complain of fever, and of course there will also be blood within the stool of the patient. Then finally, you have the enteroaggressive E. coli. It also causes a similar clinical picture to what we've just talked about, enterotoxigenic E. coli. So remember, we've said here enterotoxigenic E. coli is associated with what we call traveler's syndrome. So the same feature is also seen with this strain that we are calling enteroaggressive E. coli. So those are some of the clinical manifestations or some of the clinical features we see with somebody who is having E. coli, where the E. coli is being associated with the intestinal infection or intestinal disease. Then, apart from causing disease within the lumen of the GIT, it can also be associated with what we are calling extra-intestinal disease. So where the disease is not happening within the GIT. So, and of course, the example, the way I mentioned, E. coli can cause what you call urinary tract infection, especially if fecal matter find the way into the urethra. So some of the things you see in patients with having urinary tract infection as a sort of E. coli condition, examples of cystitis. And of course, cystitis is inflammation of the urinary bladder. So the bacteria moves up the urethra, then they find their way into the bladder where they cause inflammation of the bladder, a condition we are calling cystitis. Then they can also move further into the kidney, associated with a condition we are calling pleonephritis, where there's inflammation of the nephrons within the liver, within the kidney, sorry. So we are also saying that this is a very common feature, especially in women. And the reason why it's a common or is a frequent infection in women is just because of the anatomical position of the urethra within the women, the female gender. So remember, the urethra is just close to the skin. So if a lady does not put a lot of attention into their personal hygiene, then there's high likelihood that they will develop a urinary tract infection as a result of E. coli. And I'm very sure these are very common bacteria that has been isolated from urine culture, especially from women. So then this begs a question: what are women supposed to do? Because remember, E. coli is a normal flora, so it's a bacteria that is friendly to us, especially if it's found within the colon. So for our ladies, this tells us that when you go to the washroom, you need to be very careful, because if the water in the washroom gets its way into your urethra, there's high likelihood that you'll develop a urinary tract infection because of the anatomical position of your urethra. So the same applies when you're using the toiletries. So that is the tissue and whatever. So the movement of your tissue is supposed from front to the back. And the reason is, if you do the opposite, there's highly likely that you'll introduce fecal matter to the urethra. And of course, that will lead to a urinary tract infection. Then the other condition that is not associated with an intestinal condition associated with E. coli is neonatal meningitis. Especially this happens if the mother is having a urinary tract infection as a result of E. coli. So during delivery, the E. coli can be transmitted during this process of delivery, and when it finds its way into the bloodstream of the newborn, it might travel to the meninges and cause meningitis. So remember, E. coli has also been associated with neonatal meningitis. So that's something you need to do. When a mother comes to the clinic for delivery, then after some time the baby develops neonatal meningitis, then there's high likelihood that the meningitis could be as a result of coming into contact with E. coli. So this is something I had mentioned earlier when we were looking at E. coli, where I said that E. coli is a very common bacteria that has been associated with nosocomial infection, especially when we are doing our procedure of catheterization. So if you don't do catheterization by following the laid down aseptic procedure, remember there's high chance of you introducing a normal flora, in this context E. coli, into the urethra. And once the E. coli finds itself into the urethra, it will eventually cause what we are calling nosocomial infection. So sometimes call it CAUTIS, catheter-associated urinary tract infection. So for you as a practicing nurse, remember when you're doing catheterization, this is a bacteria that you need to put a lot of attention to, because if you don't do that, you might eventually give a patient an infection when you're carrying out your procedure of cauterization. And of course, these bacteria might eventually also find their way into the bloodstream, where it can cause sepsis or sometimes bacteremia. And of course, this is accompanied by what we call production of toxin that will eventually lead to other complicated condition. Okay, so prevention and treatment. So for intestinal diseases, the best way to handle this disease is to ensure that you are eating food that is not contaminated, or you're not drinking water that is contaminated. So the way I was giving an example of traveler syndrome, so ensure that when you're traveling, you don't eat food on the road, or you don't take water on the road, because there's high likelihood that you might get infection as a result of E. coli, especially those strains that have been associated with the intestinal condition. So because E. coli is associated with the diarrhea, then of course we need to replenish the body fluids, because when a patient is diarrheaing, of course there's high likelihood that they will lose a lot of fluid, and when they're losing fluid, they also lose their electrolytes. So for us to manage such a patient, we encourage rehydration. So we give fluids, and especially we also give a solution that they have the electrolyte, so that they can be replenished. Okay. Okay. So antibiotic of treatment, this is subjective, and the reason why we are not putting them here is just because, remember, for you to get the best drug of choice, it depends with what the culture will tell us. So after we've done the sensitivity, then the drug of choice is the one that we'll choose to manage our patient, keeping in mind that we have some strains that are resistant to some of the antibiotics we are doing. So you're saying that for you to choose the best drug of choice, the best drug of choice should be determined by the antibiotic sensitivity test. So which means we know at the sensitivity test. So whatever report we get from the sensitivity test, then that drug that exhibits the highest response is the one that we'll use to manage our patient. Okay. Do you have any question on E. coli? Any question on E. coli? Amos? Amos?
No question.
No question. We have Brenda?
No question.
Sorry?
No question.
No question. That's good. So, but remember what I want you to keep in mind. Remember here we are looking at E. coli in two lanes. So we are looking at an E. coli that is a normal flora, and at the same time we are also saying that we can have an E. coli that is pathogenic. So you need to be able to differentiate between the two contexts. So E. coli can be a normal flora, at the same time it can also be a pathogen. Okay.
Excuse me.
Yes.
I was asking.
Yeah.
Antibiotics that you're giving for this case, is it just for the bloody diarrhea, or you're giving for either?
So the antibiotic is either. Remember, antibiotic is a drug that you're using to treat a particular bacteria. So when you're giving an antibiotic, is not going to the anatomical location where the infection is. So like, for example, if you are giving a penicillin, of course, we expect the drug to penetrate into the tissues and find where the infection is. So whatever you use for urinary tract infection could be the same thing we are also using for a condition that is associated with the GIT. Like for example, somebody is diarrhea because of E. coli. So the antibiotic will still be the same.
Thank you.
Yeah. Okay. So the other group of gram-negative role that are found within the GIT, we have the salmonella. So is there any need for me to go through this? Given that salmonella is something we know?
Yes, sir.
Yes? Hello, Amos.
I think you just go through it.
You want us to go through it?
Yeah.
Yeah, but I thought this is something we manage our patient in our clinic, especially in the backyard clinic, like on a daily basis. So if somebody is having diarrhea, you will always be told that you have salmonella.
Sure.
Okay. So, but I'll just go through it briefly. So remember, it's also a group of bacteria that is found within, that has been associated with infection of the GIT. So under this species, of course, we have various species, and the one that is majorly associated with the human disease is Salmonella typhi, the one that causes enteric fever. So the one I've just been referring, when you go, you have a diarrhea, people tell you that you're having salmonella or you're having typhoid. So that is the common name on the street, okay? So we're also saying that, so salmonella are widely distributed in nature, of course. They are found everywhere within the environment, and that's why you find that they are very common. And I think that's why people are also trying to abuse. So you're having diarrhea, they say you have salmonella typhi, it's just because they are readily available within the environment. So their mode of transmission, just similar to what we looked at, that is the E. coli. So they are also transmitted through fecal-oral. So you get salmonella or typhoid fever when you eat, drink water that has been contaminated with fecal matter, and especially if you come into contact with people we are calling chronic carriers. So I'm very sure you still remember what we talked about carriers. So carriers is a person who is harboring an infectious agent, but themselves are not exhibiting any clinical symptom. So such a people are the sources of infection, especially to those people who are vulnerable, okay? So we are also saying that young children and adults are particularly susceptible. And of course, as we already know, it's just because of their immunocompromised state. So the immune system, especially for the young children, is not well developed. And for the adults, the immune system is dwindling. So, and of course, that makes them become more susceptible to having a salmonella infection. So the pathogenicity, so we are seeing salmonella invade the epithelial cells within the small intestine where they cause inflammation. And that's where we are saying that salmonella is associated with gastroenteritis. So there's inflammation within the GLT. So, but you're also saying that the disease sometimes remain localized, but it can also become systemic. So systemic is where it can penetrate through the intestinal wall, then find their way into the blood circulation and be disseminated in various organs and in various tissues, okay? So you're also saying that there are organisms that are facultative intracellular parasite that survives in phagocytic cells. So remember here, our phagocytic cell is supposed to kill the bacteria. But for salmonella, they have a mechanism of surviving. So they survive by producing enzyme that enable them jump out of their phagosome, and they remain suspended within their cytoplasm. So which means that our phagocytic cells are unable to kill them, irrespective of them being within the phagocytic cell. Okay. So some of the clinical features or clinical importance of salmonella. So we say that salmonella infection can cause both intestinal and extraintestinal infection. So when it remains localized within the lumen of the small intestine, it is causing intestinal infection. But when it gets into blood circulation, or when it's disseminated into various tissues, it becomes extraintestinal. So where it is associated with either bacteremia, septicemia, and so forth. So when it is within the lumen of the intestine, it causes a condition we are calling gastroenteritis. But remember, gastroenteritis is a general term that is given to all infectious conditions that are associated with the infection of intestinal mucosa. So when salmonella is localized, then we call it gastroenteritis, or sometimes we call it salmonellitis, and so forth. So the gastroenteritis can be associated with various species within the salmonella. So we have the enteritis, and we have the typhimurium as examples of the salmonella that has been associated with the gastroenteritis. Then, of course, we have the Salmonella typhi, the one that is very common within our environment. So some of the things you see with a patient who is having salmonellosis or gastroenteritis is where the patient will complain of having a nausea, they will be vomiting, and sometimes they will be having a diarrhea. But remember, this diarrhea is non-bloody diarrhea. And of course, it having an unbloody diarrhea we can be able to distinguish between a diarrhea associated with E. coli and at the same time a diarrhea that has been associated with the shigellosis. So remember, shigellosis gives us a bloody diarrhea. So, but we're also saying that after ingesting the salmonella in either contaminated water or food, the incubation period is around 48 hours, where the patient will start complaining of having fever, abdominal cramps, and so forth. Okay. Excuse me. Then the other condition is enteric, or sometimes we call it typhoid fever. So typhoid fever is an example of a condition that has been associated with salmonella, and especially Salmonella typhi. So, and this is an example of a condition that is life-threatening, because if no necessary measures are taken into consideration swiftly, then there's a possibility that we might eventually lose the patient. Okay. So some of the symptoms that we see with people who have fever, things like patient complaining of chills, sweating, headache, anorexia, weakness, sore throat, coughing, malaria, and sometimes even having a diarrhea, or if they're not having diarrhea, they might still complain of having constipation. So constipation is where the patient takes a very long time before they can have a bowel movement. So the complication that arises as a result of typhoid fever is where when the bacteria started causing perforation within the lumen of the small intestine, and of course, when they find their way into blood circulation, where they're associated with conditions like endocarditis. So endocarditis is where there's inflammation of heart muscle. And of course, that's why we are saying it is life-threatening. So if necessary measures are not taken into place, then there's high chance that we might lose the patient. But remember, we say that it can also be disseminated into various tissues or into various organs. So when it is in blood circulation, it causes a condition, we call it bacteremia. So it can also be associated with osteomyelitis. So that is inflammation of the bones, septic arthritis. So that is inflammation of the joints, etc. And of course, it can find itself even into hepatobiliary tract, and sometimes even in the spleen. So remember, when you're looking at salmonella, remember it is also causing what you're calling an extra intestinal infection. So it's not only confined within the GIT, but it can also be disseminated into other body structures. So laboratory diagnosis, so similar to what we've talked about E. coli. So we can say that a person is having some salmonellosis or typhoid fever if we can be able to isolate the salmonella from a stool sample. So either using a differential media or a selective media, okay? So if it is disseminated, then we can do a blood culture or sometimes a bone marrow aspirate, depending with the tissue that is being affected. So once it's not in the GIT, it is in circulation, then we no longer use stool as our sample of choice, but we do a blood culture. So from this blood culture, we are able to isolate the salmonella, okay? Treatment and prevention. So for gastroenteritis, especially in immunocompromised hosts, so we give antibiotic therapy. So remember here, we are not even giving a specific antibiotic, because the issue here, we need to give antibiotic based on the sensitivity test, okay? And that's why we encourage, when you're suspecting a patient to have a salmonella, you don't just give antibiotic, because there's highly likely that the antibiotic that you're giving, the salmonella could be resistant to that given drug, okay? So prevention, of course, the prevention is the normal thing, the way we just talked about E. coli. So we are saying that for us to prevent typhoid fever or salmonellosis, then what we need to do is to get rid of fecal matter. So by ensuring that we have proper disposal of human waste, correct handling of food. So we ensure that our food is not getting contaminated with fecal matter. Then, of course, ensuring what you're calling good personal hygiene. And of course, it's the same approach we are using to try and control the spread of COVID-2019. So we are telling people to wash hands, ensure that they are cooking their food well, and that in itself will always try to help us try to prevent the disease. So apart from hand washing, ensuring that you are cooking food well, we can also prevent salmonellosis by giving a vaccine. So there's a vaccine that we can give people, especially those people that are vulnerable. And of course, this vaccine, what it will do, it will go and initiate an immunological response that will protect an individual from getting the salmonella. And I'm very sure most of us are familiar with this vaccine that we currently use in our clinical practice. Okay, so that is the summary on salmonellosis. So we're saying that they are facultative anaerobe, they ferment glucose, and so forth. So these are just the culture characteristics. How do you differentiate between salmonella and E. coli, given that we are using the same sample, that is a stool sample? So these are some of the features we'll see with a colony that is coming from salmonella. Okay. So do you have any question? Any question?
I have a question.
Yes, Brenda?
Because you say that we have to use an antibiotic based on sensitivity.
Yes.
Is that in blood or in stool?
Sorry?
Should it be in blood or in stool?
No, it's just the same thing I was telling. I don't know who was asking the same thing. Remember, the antibiotic is targeting the bacteria, but it's not targeting the specific organ that is being infected, where we have the infection. So like, for example, if you are giving ciprofloxacin, so remember most of the antibiotics that we currently use are broad spectrum. So broad spectrum is content that they can be used in various conditions. So if you're having an infection with the GIT, when you give ciprofloxacin, it will manage the same. Like, remember, we are saying that when you're having a local infection, it can eventually be disseminated. So you're having still the bacteria within the colony, but at the same time, the infection is still within the tissue. So when you're using the antibiotic, we are not selecting the antibiotic based on the site of infection. We are choosing the antibiotic because of the bacteria in question. So like, for example, if we are using ciprofloxacin, of course, it's just because we've isolated the salmonella from the patient, and our antibiotic that responded by killing the colonies on an artificial media was ciprofloxacin. And of course, we expect when you give cipro, it will have the capability of penetrating in the entire system of our human body.
My question is,
Yes?
for you to choose the antibiotic,
Yeah?
you have said we need to base it on sensitivity.
Yeah.
So which sample are we taking, blood or stool?
So the issue is the context. Like, for example, if it's still within the local environment, what will be the most appropriate sample?
I'm going to assume stool.
Yeah, stool, because it's still within a local site. So that tells us that it's still within the GIT. So once it gets disseminated, what do we go for? Do we go for stool?
I guess you'd go for blood. But
Yeah.
now if patient comes in with sepsis, with you having no history of, you can do both. You'll find it in.
Yeah, you can do both. Like, for example, there are those signs you talked about. We have the symptoms that a patient will exhibit, especially if it's within the GIT, okay? Then we also say that there are those symptoms the patient will exhibit when it's extra-intestinal, so which means the infection is not within the intestine. Like, for example, if it's out of the GIT, of course, you'll expect a patient to be constipated. So if a patient is constipating, of course, the patient is not diarrhea, okay? But another patient will come and is having a diarrhea, then of course you suspect the patient to be having, for example, typhoid fever. So the best sample for that context, then you need to go for, for a stool sample. But you see, a patient is constipating, then there's a likelihood, in fact, they will not be in a position to give you a stool sample. So what do? You can request for a blood culture. So, and of course, in that blood culture, of course, you are trying to address in an event where the infection has disseminated. So it's no longer within the lumen, but it's spread into the tissue.
All right.
Yeah, so of course, that's why it's very important for you to be very careful, especially when you are taking your clinical history. And if you don't get your clinical history very well, then there's a possibility that that event can lead to misdiagnosis. Okay, another question? Nancy? Nancy?
Just a minute, please. Pass.
Okay. So any other question?
I will come up.
Jen?
Yes?
Any question?
I'm okay, thank you.
Yeah, okay. So I think we'll stop there.
Before we stop, mine will be an addition from the question from Brenda, the one you have asked. She have asked about the type of specimen that you take. I think type of specimen is determined by the patient presentation.
Yeah. Then
the locality of the facility, because if you are in a local facility like a healthy center, of course you'll have the simple specimen like the stool. Then it's go scaling above according to the facility that you are. So then you give the antibiotic that is sensitive to the one you maybe have isolated from the specimen.
So, but remember here we're saying the isolation is based on the availability of facility. Like, for example, in a health center, remember all these things, our laboratory tool that we can use without contradiction is a culture. But of course, remember, I've just talked about people being misdiagnosed of having salmonella, which is a very common practice. I don't know whether you people are practicing the same. And of course, that led to, remember there was a serological kit we were using, the one we were calling widow kit, So people were misusing it. So in the event you can be able to do a stool culture, and then of course we we expect you also to be able to do a blood culture, because remember, all of them are culture technique. So, but here the aspect is just the clinical presentation. What is the patient presenting? So if he's presenting a condition that you suspect, there's a likelihood that the condition has disseminated. Like, for example, you'll ask yourself, a patient comes to the clinic and he tells you that he's having arthritis. So we say that arthritis is an example of a complication in salmonella. So of course, you'll try to try and rule out. So you ask the patient whether he had some experiences of diarrhea. Then the patient will tell you that like 2 weeks ago, he had some diarrhea, he had some chills, he had some fever, but that eventually disappeared. So what comes into mind? So you will highly suspect that this could be a disseminated salmonellosis. So that's why I'm emphasizing, especially for a clinician, history taking is very, very, very critical, because if you don't get it right, then there's high chance that you'll eventually end up with a misdiagnosis. So remember, I've said that salmonella is an example of a bacteria that causes enteritis. The same applies to Shigella. The same applies to some virus. So how do you distinguish? So you can only distinguish either by having a comprehensive clinical history taking, clinical examination, then of course with an eventual of where you're able to isolate the microorganism from a sample from the patient. But before you know which sample to send for, you need to have gotten it right from your clinical examination. So you need to know all those manifestations, digging back into the past, then with an eventual guide to know which is the most appropriate text. So like, for example, if you assume that salmonella only causes diarrhea and a patient comes to your clinic is having disseminated infection. So remember, if you are basing your diagnosis on diarrhea, you will eventually lose a diagnosis on those patients who have disseminated infection. But remember, we said that this is a life-threatening infection. And that's why you find that if your patient is not managed well, by the way, if you're having salmonella, you can eventually die, especially if it becomes complicated. Is that right? Harriet? Harriet?
I'm okay.
You're okay.
Yeah.
Okay, so I'll make one request. I don't know whether we can be able to read on the remaining microbes. Then maybe next week, we just review them so that we move to the next unit on virology. Is that okay?
Okay, it's okay.
Yeah, so let's just review following the sequence in our discussion. So those places that you find you are having challenges, just write them down. Then maybe when you have the presentation next week, we can highlight those challenges. Then we move to the unit of virology.
Okay.
Okay. Maybe an announcement. Has everybody gone to Moodle?
Yes.
Yes.
So what is on Moodle? Kim, what have you seen on Moodle?
Oh, there was an assignment that was sent yesterday.
Yes?
There was an assignment that you sent yesterday, and
Yeah,
due today at 9:30.
Yeah, so that is a quiz to summarize our unit on bacteriology. So
Okay.
I'll expect you to take the quiz. So, but remember, just take your time because it has 2 attempts. Okay?
Yeah.
So remember, because we are unable to come to class, whatever we are doing online, that will form part of our continuous assessment. So ensure you are capitalizing on that. Don't do it in a rush because I don't want you to fail. So it's an opportunity, read and choose the correct answers.
Okay.
Okay. Another thing you've seen on Moodle?
I've not seen anything else. Nothing new. In microbiology, I have not seen anything else.
So remember, currently I've incorporated new YouTube videos. Have we seen the videos?
Yes.
Yeah, so, yeah, so you just take your time. At your free time, if you are next to a Wi-Fi, you can just listen to the videos so that they can continue strengthening your learning. Is that right?
Yes.
Okay. So if there's no question.
I have a question.
Yes, Harriet.
Like the last quiz that we did online, there was a question 5 people answered according to how they understood the difference between bacteriostatic and bactericidal drugs. So with this same cut wheel, are we supposed to answer? Is it like computer generated the answers? So that if someone answers the way they understand it, it's still correct, that they will not get it wrong.
So is it a multiple choice question?
Yes. No, it was like I'd state the difference between bacteriostatic and bactericidal drugs.
Yeah, so remember the online in itself is a mixture, because remember there's a truth, there's an MCQ, there's also a matching whatever. Is that true?
Okay. Yeah.
So what you do, of course, I want you to distinguish between a bacteriostatic and bactericidal using your own words.
Okay.
Yeah. So once you do that, I'll be able to see it from my side.
Okay.
Yeah. Another query?
Yes, another question.
Yes.
I want to take you back to Neisseria meningitis, whereby we say that we can use these vaccines which we are giving between 11 to 55 years. You called it conjugate. What did you mean?
Oh, so they call them conjugates.
Yes.
Somebody to answer that? Especially those people who are giving them. We need to get answers from them. Hello? Somebody to answer. Is it Jedit or who? Diema?
No, Mercy.
Mercy.
Yes.
Hilda? Hilda? Samuel?
Just a moment. I'm trying to remember what exactly it was, because it is
So I'm very sure we are giving the meningitis vaccine in Kenya. That's true. So what are we telling our patients? Especially when they read the insert and it's being called conjugate.
Can I try?
Yes, Caroline.
Is it due to contact?
No, it's not contact. Diema? So the word conjugate is an English word, isn't it? Conjugation.
a combination of two.
Yeah, conjugate, you take two things and put together. So when you're saying a vaccine is a conjugate, which means it's having two components, okay? Yeah. So for it to be effective, sometimes we have a carrier molecule, and the carrier molecule, sometimes we call it a vector. So because we want our vaccine to be effective, we can couple a carrier molecule with our target structure that we want our immune system to respond to. So when we couple a carrier molecule and our vaccine candidate structure, then we eventually end up having what we call a conjugate. Is that clear?
Yes.
Yeah, so a conjugate is having two molecules that are totally different to each other. And majorly the reason why we have such a vaccine, remember, for us to have an effective immunological response to a vaccine, then we need to use structures that will adequately stimulate the immune system. Because if the immune system will not be stimulated the way it's supposed to be stimulated, we'll eventually have a weaker immune response. And it's the same thing we are talking about the COVID-19. So that's the discussion they're asking. People asking whether those people who are recovering, they have a long-lasting immunity or is a temporal immunity. So, of course, the factor behind all these things is it's dependent on what arm of the immune system is being stimulated. So if you are able to stimulate the cell-mediated arm of the immune system and the humoral together, then our immune system is long-lasting. I'm very sure, yeah, in the vaccination, remember, we looked at advantages of live vaccine and disadvantages of killed vaccine.
Yeah.
Do you still remember that? So we said that killed vaccine will give us a shorter, the lifespan of the immunity is short. And that's why we are encouraged to give booster dose. So we are giving booster doses just because killed vaccine gives us a weaker immune response. Okay? So for us to overcome that, mostly we use what we call conjugates. So we'll take a molecule and then couple it to our vaccine. Then the conjugate, the other molecule will stimulate a stronger immune mechanism. And that's why we call them conjugates. So it's just the same way we talk of polyvalent vaccine. Or sometimes we call others combined vaccine. So it's just the same thing, looking at our vaccine preparation. Another query?
Yes, I had a question.
Yes, Irene.
Yes, I had a question on E. coli.
Yes.
I wish you clarify how we know that E. coli, they are normal flora in a normal circumstance.
Yeah.
But then we got these other, they become pathogenic. Maybe you help us to connect how they live to be the normal flora to become the pathogenic. Yes, how those factors that make them to be pathogenic.
Somebody
Are they host related or they are the E. coli themselves related factor?
Somebody to answer that, Irene? To answer Irene? Is it Irene who is asking?
Yes, please.
Who can try to answer Irene?
Can you give the question once again, Irene, please?
So is
yes, I'm asking E. coli, they are normal flora on a normal circumstance.
Yes.
But then now we have learned that they can still cause, they can become pathogenic.
Yes.
So my question is, what are the factors that make that transition from normal flora to a pathogenic microorganisms?
I think, as Festa said, normal flora is normal when it is located where it's supposed to be. But once it migrates to another place, that's when it brings diseases. I think that is the factor. The first factor is migration to another place.
No, it's just like the same thing. I don't know, I can use which example. So it's like you're living in the house. You have a house, you have a bedroom, you have a kitchen, and of course we have a regulation. Okay, so you have a neighbor. So of course you stay. If your neighbor is a good guy, is a good lady, but when you wake up one morning and you find him in your kitchen, is he your neighbor?
No.
Yes?
No.
Yeah, so remember, everything has boundaries. So he's your neighbor when he's in his house. But when he moves from his house and he pitches comes in your kitchen, he's no longer your neighbor. He's become an intruder. So remember, there's a balance. So E. coli survive within the lumen because there's an interaction and there's a normal understanding between E. coli and the immune mechanism within the lumen. And of course, there's a way the body protects itself, looking at what we learned about all the 3 levels of defense mechanism. So if E. coli finds its way into blood, so remember, it's going into a place that it will flourish. And of course, that is the intention of every microorganism. So once in a new environment that is favorable for it, it will start multiplying. So as it starts multiplying, it will eventually start destroying those tissues. So it's a normal flora within the lumen of the intestine, because our body has a mechanism to deal with it and to ensure that it's not crossing the boundary. But when it moves from the lumen into blood circulation, so remember, within blood circulation, we don't have a mechanism to tame it. So the way I was just talking about your neighbor, when he comes into your kitchen, apart from him being your neighbor, you know him, he's a good person, Nene, but he'll eventually start feeding on your food. So when he starts feeding on your food using your utensil, what happens? He becomes a parasite. That's true.
Yeah.
So that is the context. And remember, when we were looking at normal flora, we said that normal flora only are found within the external surfaces. That is on the skin, in the mucosa, in the ears, in the nose, those environments that are exposed. But within our deeper tissue, we have microorganisms. And the reason why they live on the surface is because, remember, our first barrier is the skin. So they cannot penetrate the skin. But when they go into other environment where maybe we don't have the skin tissue, so you find that the bacteria can eventually start causing damages within those environments. So when you're looking at any normal flora, that is the context you need to be looking at. Be it E. coli, be it the Staphylococcus species. So those are the contexts we are looking at. So they're only normal flora if they are supposed to be where the body can manage them. When they change that environment, then they eventually take advantage and they start flourishing. So, and in fact, when you're looking at normal flora, it's not only in a healthy individual. So if you become immunocompromised, where your immune system is unable to protect you, what will happen? The normal floras are the ones that will start causing disease, just because of the state of the immune mechanism. Is that clear? Yes.
Thank you. Question. When you talked about the reservoirs and the vectors, vector is the disease-causing agent, e.g. the mosquito, right? I was going through this, I was going through rabies. This is when it comes to rabies. Is the dog a reservoir or a vector?
Vector.
The dog, is it a reservoir or a vector? So one thing we need to do, can you define a vector? What is a vector?
There's a vector.
A disease-causing agent.
No, it's not a disease-causing agent. Who can answer, Caroline? Anne? Oppo. This Oppo should be Jen, yeah? Jen? Jen? Samuel?
From what I understood, is that a reservoir is like the storage of the disease-causing microorganism. Oh my goodness! So that is what I understood. But for a vector, is the A vector is the introducing part of it.
Okay, that's a good time. So remember, a vector is a microorganism that aids the transmission of a disease-causing agent from one host to the other. So if you are saying that a mosquito is a vector, a mosquito will carry a malaria parasite from patient A to patient B. Okay? So, but for a reservoir is a point or is a place where people get the infection from. And remember, when we are looking at reservoirs, we say that you can be having a living and non-living reservoir. So a living reservoir could be, like, for example, a person who is sick for salmonella. Like, let me just use an example of COVID-19. So why are we tracing these people? We suspect they've come into contact with these people. It's because these people are acting as source of infection to more other people. So for those people who already have the infection, we can term them to be reservoir hosts. The reason we are calling them reservoir hosts, because if they come and speak where you are, they come and shake your hand with their contaminated hand, they eventually give you the infection. So a reservoir acts a point where people get the infection from. So the way I've said, it can either be living or non-living. So if it's non-living, for example, you are a nurse, you are using surgical equipment that are not sterilized. So what happens? So the surgical equipment will harbor disease-causing agent. So as you use those surgical equipment on another patient, you find that you eventually give the other patient an infection. And that's why we are encouraged to do sterilization. So sterilization is a process where we're trying to get rid of all forms of microbes from our equipment. So a vector is used to transmission. A reservoir is where susceptible hosts get the infection from. Is that clear?
Yes.
Yeah. So remember to distinguish between the two. One is a vessel, the other one is not a vessel.
So it's okay to say that a reservoir is a source of infection?
Yeah, a source of infection. So that's where people get infection from.
Okay.
Yeah.
The dog is a what?
Another query?
I had a concern. Kindly, is it possible, like for the deadline for the submissions today?
Yeah.
You pushed them a bit forward, because of timelines. Like for instance, we might be having a class of epidemiology and anatomy. And personally, I'll be working on night shift. So how can you assist me on that, or probably another person who is in a fixer of that manner?
Oh, so you want an extension until tomorrow morning?
That would be better if it is possible.
So I'll revise it and put it tomorrow morning.
Thank you.
Okay, welcome.
I had a request as well.
Yes, Brenda.
Could you kindly help us with revision questions? Like from previous exams, because now we can't really interact with previous classes. We can't borrow their past papers for revision use.
So why are you so interested in the revision question?
Because that helps me read the topics. So
but
that
there's a revision section on Moodle.
Yes?
You've seen the revision part of Moodle?
Where?
Yeah, so if you go there, you see some of the sample question. But to me, most of the time, I will not encourage people to use revision question to do their revision. Because remember, what most of the time I will assess you is what we are discussing. Like what we are talking now, we are interacting, we are asking questions. You see, to me, that is more meaningful than saying that we'll use past papers to do our revision. So what I will encourage everybody is to actively listen to what we are saying, make a contribution to what we are saying, because this is actually where we'll get our assessment from. So we want to assess people on actually things that we've been doing together. So things that everybody has participated actually. Is that okay? But if you still want the question, you go to the revision section to find those questions. Is that clear, Brenda? Brenda?
Where is the revision section?
The revision section should be shells, should be 7 or 8. You can see it?
Where?
So if you go to your dashboard and look at the units we are covering, there's a place we say the revision should be revision section, revision something.
Okay.
You've seen it?
I'll look for it.
Okay. In fact, before I forget, your question has just triggered something I'd sent. So there's something I'd sent on email. I don't know whether you saw it.
The one for
self-evaluation.
Yes.
So have we done that?
Yes.
So ensure you're using that as a guide to know where your strength is and know where your weakness is. So after you've scored yourself, because remember it was asking you question, then from there it will tell you where you fall.
Okay.
Because self-evaluation is a very, very important thing. Every time you do whatever you do, you always need to be evaluating yourself. Are you grasping the concept? Are you struggling? Something like that. So of course from there you know where do you need to put a lot of effort. Okay. Okay, so if there's no another question, I think we'll stop there.
Thank you.
Okay, welcome.
Thank you so much.
Class announcement. Hello.
Hey, Sema, Kim.
Oh, class announcement. Kuna like daily announcement that I have. Kuna moja ya epidemiology class will be 1 up to 2.